He kōna e purangi tēnei nā te reo irirangi o Aotearoa.
Are you shy? No, you're not shy. Say hello to Claire. Say, this is a Radio New Zealand podcast.
James Espinas-Ray is only 4 months old. So introducing the podcast might be a bit beyond him. But already he's well able to perceive things around him. To spot movement and contrast, and especially faces.
Pretty early on, he quite liked looking at the little purple monkey in his little activity gym. Has a little smiley face in it. Basically just a lot of faces.
Yeah, it was kind of crazy that even a very abstract face, which was this little monkey toy's just got 2 dots and a little curve for a smile. And he was obviously way more interested in that than he was in anything else that you'd dangle in front of him. It was kind of crazy.
Kia ora, nau mai haramai ki te ao hurahanga, ko Clacken Callan tēnei. If you're a Black Sheep podcast fan, you might have already recognized that voice.
I'm William Ray, I'm your colleague, I work with you. I produce programs for Our Changing World sometimes.
And I'm Janella Espinas. I'm William's wife and James's mum.
Today, we're exploring eyesight and visual perception, and we're starting at the very start.
In some ways, the question is, why is the sensory system in a certain state at birth?
Professor Vincent Reid spent 25 years of his research career investigating how infants learn about the world. An important part of this is how infants' perceptions of the world interact with and influence how their brains work. But he realised that he and others in the field were working off assumptions.
Actually, there are a whole lot of suppositions about the nature of newborns and neonates in terms of what capacities they have, how they have them, why they have them. Much of that was based on no actual data. And so that's when I started looking at the human fetus and realised that we really didn't know very much at all about what was going on in the 3rd trimester of pregnancy, specifically when you had sensory systems that are operational, but at the same time, we didn't even understand the environment in which they were processing information.
When babies are born, they've a certain amount of ability that then develops rapidly as they interact with the world. Here's Janella and William again, on when James first looked at them. Now, James arrived a bit early, so he spent a few days in the NICU before heading home fit and healthy.
He was pretty sleepy at the time, but every now and then he would wake up and, you see photos of what babies look at or what babies' vision is like on the internet, and it's all pretty blurry, but I like to think that he locked eyes on me. It was pretty cool.
I think he definitely did. Yeah, I remember certainly the second day. Were you right? Certainly the second day, we sort of I got to hold him for the first time, and he was definitely looking at me from sort of short range then. He couldn't pick you up from across the room like he can now, but he could definitely, when you were right up to him, he looked you straight in the eyes, yeah.
And this locking on to their parents, says Vincent, is something that babies are really good at.
Newborns, for example, are very good at detecting movement in the periphery, and that's one of the main things the periphery does. But in terms of focusing on things and getting information in detail, very bad. Yet a newborn is, within the space of a matter of 2 or 3 minutes, can actually individuate a parent from a stranger. So in other words, very rapid learning occurs in the visual domain for things that really matter. So how and why that happens is a set of open questions.
But Vincent's question was, what about before they're born? What do they see, if anything, in the womb? And is this already shaping their experience of the world?
How does perception actually begin to even start? What are the building blocks of psychology which then come from perceptual mechanisms?
Vincent is a developmental cognitive neuroscientist, interested in how human brains develop, how our sensory systems develop, and how this relates to our understanding of the self and others. So, big stuff. Stuff that gets to the very heart of us as human beings. Our sensory systems are how we perceive and interact with the world. They in turn shape our experiences and our sense of self. And sight is a really important input.
Vision is just so incredibly important. for us as humans. We're a visual species. The auditory system is very important, but actually so much information we get from the world is from vision, mainly because you can tell what's going on near you, you can tell what's going on far away from you, and that will inform your future actions and your future behaviour. So vision tells us so much, and it's also so complex. It's really not actually one sensory system. It's a collection of different parameters that we merge together and we believe is one thing. To give an example of that, your acuity, your ability to actually focus on something and see it in high definition is only a very small part of the visual scene at any given time. Yet, most of what you're processing is in your periphery. The stuff you see in the periphery is actually very rough, it's very raw, and it's actually also processed in black and white. And yet you fill in the colours yourself through tricking yourself that there are colours there, but your eye literally cannot see the periphery in colour.
This is because this complex visual system involves not just light bouncing off things you see and hitting the back of your eyeballs, but also incorporating pre-existing information that you already have.
You have two different mechanisms. You have bottom-up information that comes from the environment through your eyes and into your brain, and then you actually have your brain telling you what you're seeing. So top-down, cognition affecting perception. And so you know that there are colours in the world, you know that things should be certain colours, you may have seen them previously when you've been oriented in a slightly different angle in the world, and therefore you fill in those colours in a way that kind of makes sense, and you do it effortlessly. And that effortless aspect is the bit that makes vision so fascinating. How do we do so many complex things without even thinking about it, without even knowing that we're doing it?
And when you say that top-down bit, that, from being out in the world and knowing that colours exist, that my brain is filling them in, how do we begin life then?
Now that is the question, and that's one of the main reasons why I've got so interested in this research topic in this area.
That may be the question, but there's another more basic question to ask first. Can fetuses even see anything in the womb? To answer that, Vincent enlisted help.
I'm Jacob Herrikhausen. I'm an associate professor in mathematics, and so I do mathematical modelling of actually all sorts of different things.
Things like how particles from interstellar space move about in our heliosphere, the area of space impacted by wind from our sun, which, is quite different to what Vincent asked him and his PhD student Zach Isaac to model. There are plenty of layers between light and the fetus in the womb. Clothes, fat, muscle. So is anything getting through?
The main conclusion of Zach's PhD is that, yes, there are, under fairly realistic configurations of those layers, we do expect a reasonable amount of light to get through to the uterus. I guess the main conclusion was that the level of light is comparable to a night sky with a full moon. So certainly when I go outside now every time and there's a moon, I'm like, oh yeah, this is like the amount of light that gets through to a fetus. It's significant.
And had nobody looked at this before?
Not in this much detail. So people had looked at different layers, but we added additional factors like curvature. Also, most people have some sort of layer of clothing on them. So different things that limit the amount of light getting through.
In fact, the leading model had been developed by shining light through dead chicken breast, says Vincent.
Which is nothing like living tissue and nothing like all the different types of tissue that are in a human. We actually had all of the optical properties in the model related to skin and adipose, muscle, the uterine wall, and then through to the amniotic fluid. We had to really hunt around to find some of these properties actually in terms of the scattering and what actually happens with light when you shine it through these different tissues. But we found them, they're in the literature, and we put them all together to allow us to build the model in a way that was realistic.
So their model predicts how light will bounce, scatter, and pass through all these layers, and how much makes it to the inside of the womb. Different colours of light travel at different wavelengths. And white light and sunlight are made up of a mixture of these colours. As they explored this, they found that red light was getting through more easily than the others.
Blues and greens didn't penetrate far enough. It was only in the red area of the spectrum, which means that if a fetus is having some kind of visual experience, it's going to be with red light.
So, a red moonlit night. Of course, it's all very well knowing that light is getting into the womb and wanting to study the visual abilities of a third trimester fetus. But the next question is, how?
What we did is we used an ultrasound. So there are different ways to use an ultrasound. You can do a two-dimensional ultrasound where you basically get a slice through different tissues. And this is what maybe most people know that an ultrasound can do. If you do that and you look at the eye of a fetus, what you have is a big sort of round black thing. It doesn't look like much, but around the edges of the eye, you can actually pick up the lens of the eye. So the lens shines very brightly. You can see it very clearly. And so if the eye moves, then you can actually pick up where the lens is, which means that you can tell where the fetus is looking.
Wow, that's incredible. So you can, using ultrasound, this 2D ultrasound, you can see where a fetus in the womb is looking.
That's exactly right. And that's very powerful because so much of infancy research is about where is the infant looking and what are they looking at and why are they looking at these particular things. We can take much of those methods that have been used for many decades in early developmental research and apply it to the fetus.
He brings up an ultrasound example for me to look at.
At the top of the screen, effectively you have your ultrasound probe and it's going through skin and then muscle, adipose, uterine wall, and then at the bottom you can see this clear area which is actually amniotic fluid surrounding the fetus. And the fetus is the thing we're really interested in here. This is a focus on part of the head, and the part of the head we're interested in also happens to show us the eye, which displays as a big black circle.
Oh, I see it.
Yes. And
Right, now that you have said that, I had not realised what I was looking at. Okay,
Yeah.
so the top are still images and you can see the layers of all those different tissues that you talk through.
Yes.
And then a darker region, which is the fluid, and then the fetus in there. But below is a video and you can see movement, including that big round dark eyeball.
You can also see that there is quite a bright disc which reflects the ultrasound. So what we have here is we have a fetal eye that's rotating in the socket. You can see it moving up and moving down, and that's because a stimulus is being presented that is going up and going down, and the fetus is moving the eye in order to track that particular visual stimulus.
So this is a kind of video that you would take as part of an experiment and then would be
Yes.
analysed.
That's right. So this is the raw data. We then have to put it into a coding framework, which is called Observer. Then we sort of look at it frame by frame to see, is the eye moving? If so, where is it going? And which direction?
It's totally fascinating looking at this image in video. And that's part of why they don't struggle to recruit study participants, says Vincent.
What tends to happen is that actually someone comes in, does the study, and they tell their friends. And people who are pregnant tend to know a bunch of people who also happen to be pregnant. And they say, this was really interesting. And then suddenly we have a bunch of people saying, I'd like to do your study, please.
From a participant point of view, what's involved for them?
Well, so they come into the hospital, it's after hours, they're greeted by a PhD student who will say hello, welcome them and explain the study, what's going to happen to them. They then have an ultrasound where we actually look at the anatomy of the fetus to make sure that everything is fine, and once we've done that, we then do the study. We take a photograph or two of the screen so that you can see your fetus, you can show your friends when you get home, and we say thank you and goodbye.
Do you feed back to them the different results that come out of the research?
Oh yes. As soon as a study's published, we then contact all the participants and say, this is what's happened and this is what we now know that we didn't know before.
The first study they did was published in 2017. So before Jacob and Zach's modelling demonstrated that light could get through to the womb. Vincent started with a first basic question.
Would the fetus look towards the stimulus, or would they actually move their head away from it? And overwhelmingly, fetuses would actually look towards the light stimulus they obviously found it interesting or they wanted to engage with it.
So when you say the light stimulus there, I mean, are we talking about a dark room and you're shining a torch on somebody's belly?
In a way, that is what we're talking about, I suppose. So we're in a hospital. There is an ultrasound going, so we can see what's going on. The light levels are lowered, so they're not the same as if you're outside on a sunny day. And then what we have is we have a light stimulus, which is actually a red light. We aim it for a certain location, and the certain location is actually the periphery of the fetal eye. There's a reason for this, and that's in all the infancy research. If you present a stimulus centrally, often you get no effect. And by no effect, I mean, let's just say you show a happy face and a sad face to a newborn. It appears that they don't spot the difference. But if you present the stimulus in the periphery and they actually saccade with their eyes and engage with it, then you can move the stimulus away, further away into their periphery, and you can track how long they will look at it. And that's a nice measure of engagement. And so we wanted to mimic that with our fetuses to see whether or not they would have similar parameters of visual processing. And the answer is yes, they do.
And while this doesn't sound all that surprising, that a fetus in its third trimester would have similar visual abilities to a newborn, Vincent says it was something that just had never been checked.
It makes the most amount of sense. But there have always been people in the field who have said much of what infants do is a function of very rapid learning that happens at birth. There is something known as filial imprinting. So the classic example of filial imprinting is chickens hatch, the first thing they see is the farmer, and they will all follow the farmer thinking that the farmer is their mother. So that's the classic filial imprinting example. And so there was a large number of individuals saying that much of what newborns do is due to very rapid learning after birth. We were interested in seeing whether or not much of what newborns do is a function of capacities they already have during the fetal period. So that rules out postnatal experience as an explanation for why our visual system is structured in certain ways.
As well as shining a light centrally and at the periphery to see whether the fetus responded differently, Vincent and the team also checked some other aspects of the visual system seen in newborns, such as anticipation, which is responding to a cue and looking towards a light source before it switches on. His former PhD student Jessica Leo did these studies.
So you might think that anticipation is very complex. It isn't really. It's understanding that there is information in the environment that you can tell will appear at a certain time. So her study was very simple, very Pavlovian. You present a sound, you then present a light. You present a sound, you present a light. You present a sound and no light. And you see, will the fetus actually look to the location where the light is in the absence of a stimulus? That means that they're anticipating that the light will be there. And so she found that this is also what happens in the third trimester.
So that's like when you're saying the sensory system involves all kind of different aspects that are pulling together. When you say the cognitive side, this is the brain, like this is some kind of link in from the brain that's enabling this anticipation.
That's right. So anticipation occurs when you have a basic memory trace, when you know that's what's happened recently and what therefore you're building up statistical regularities of what's likely to happen in the future. You're making inferences, you're making predictions about what will happen. That makes it sound very complicated. It's actually quite simple. Other animals, other species can do this that are not complex vertebrates. So to say it involves the brain, that involves cognition, that still doesn't mean that it's higher order cognitive functions. It just means that the fetus has the capacity to detect and interpret and remember certain forms of information for a limited period of time that allows them to make predictions about the future.
Another study they did involved 3 dots in a kind of upside-down triangle configuration.
Schematically, that's actually very similar to the way that our faces are constructed.
Again, this is linked to an ability that newborns have, like James Espinas Ray. And Vincent wanted to see whether it was present in the third trimester.
If you show a capital T to a newborn and you move it in the periphery, they will track that more than if you show an inverted capital T. So the thing that newborns find interesting about human faces is that it happens to have more information in the upper than the lower component. Your eyes and eyebrows, there are more of those than that you have of a nose and mouth. So it's that overall configuration that draws the attention of the newborn. My first study was presenting 3 dots of light that resemble that same schematic, and then moving those in the periphery to see whether or not the fetus would turn their head to pay more attention to that when contrasted to the same stimulus but inverted. And what we found was that the fetuses would preferentially engage with a stimulus that was in that face-like configuration.
That's wild. Because they've not seen a face.
No, they've not seen a face, which means that the thing that drives an infant's interest in a face is not the face itself, it's the way that it's structured. And that's why I wanted to understand what's going on in that uterine environment and what is it even like, how much light is in there. And so the work with Zach Isaac and with Jacob, what that showed us was that light is getting through, and that light is probably enough for most fetuses to have a visual experience. The reason why that's interesting is because what is that visual experience and how is that shaping vision? So I can immediately think of some things that matter there. One thing that matters is the formation of a rudimentary body schema. So if you've got a hand in front of your eye and you move the hand and it's lighter, you move the hand and it's darker as a function of where this part of your body is, then that means that you're going to be getting feedback from your own bodily movements, visual feedback, that tells you where that part of your body is. That means that the fetus, if they have a visual experience, it's likely to help form the way that the fetus understands their own body and their own body in space.
So visual learning is starting in the womb, which is totally fascinating to think about from a psychology point of view. But I ask Vincent, how does this help us practically?
One example is what do we do in neonatal intensive care units. So you've got premature infants who are born before time, before term, before really they should be born, and they're put into a neonatal intensive care unit. I was in the neonatal intensive care unit the other day. The auditory environment, the visual environment are entirely unlike what a fetus should encounter. So you have downstream effects for premature children that are a consequence of whatever caused prematurity in the first place, but also the fact that all the sensory systems are effectively overexposed too early on in life. So this work can actually inform what those units should look like, what they should do. I think that it's an incorrect assumption that the environment should be as quiet as possible. If you think about the auditory environment of a fetus, it's actually quite loud. You've got maternal digestive system going on, you've got maternal heartbeat that's always there. Then there's maternal voice. So the fact that that's generated internally, that just goes right down to the fetus and is actually quite loud. So the idea that it should be very, very quiet in there makes no sense. The idea that you should have people talking at normal volume also makes no sense. So what is the correct parameters that would actually mean that those children have the best likelihood of not having ongoing developmental effects because they've been overexposed or underexposed to stimuli? The same is true in the visual domain. We know that there should be light, so it shouldn't be just no light at all. Equally, it shouldn't be light under ordinary lighting conditions in a hospital. And if you go and look in one of these neonatal intensive care units, then it's actually going to be completely out compared to what the child should be encountering. So those parameters should inform us what best practice should be like in those environments, which then in theory would lead to downstream health benefits for those children compared to what we do with them currently.
Vincent mentioned the sound environment there, and his team's current research is looking at the auditory world in the womb, and in particular, focusing on how the fetus interacts with music. For Vincent, this work has answered some questions, but as often happens in science, has resulted in many new ones too.
This has opened an entire door to a new field of psychology. Now we can start looking in this critical period of development. So for me, There's a whole suite of studies that have happened with newborns and with infants, which we can explore with a fetus to further understand the parameters of the visual system and what are those basic building blocks of sensory systems that the fetus has, and then how does that cascade to later forms of information that can be acquired by a newborn child.
Meanwhile, Janella and William are just enjoying watching James learn.
Now it's like he can, like, as soon as you walk in a room, he snaps onto you, and like if you smile at him from like maybe not really far away, but from like, you know, the other side of the room, he'll smile back at you, and like he can obviously pick you out and what you're doing. It's very cool to watch him change. It's very cool. He's very cute.
Thanks to Janella Espinas, William Ray, and James Espinas Ray. Thanks also to Professor Vincent Reid and Associate Professor Jacob Herrickhosen of the University of Waikato. Jacob talked about the work he and PhD student Zach Isaac did. For more about the innate music ability of humans, including toddlers, you can listen to the episode we did called Our Musical Minds with Professor Sam Muir in 2023. I'll link that in the show notes. This episode was produced by me, Claire Kincannon, and sound engineering was by Phil Bench. Script editing was by Miranda Fisher. Tēnā koe I whakarongo mai. Thanks so much for listening. I'm Claire Kincannon. Have a great week. Kia pai, te wiki.